Good. Good afternoon, everybody. Hope you had a lovely lunch and enjoyed the right amount of sunlight that you required. This afternoon we're switching gears a little, starting off with a bunch of talks relating to various genomics areas, and then following up in the afternoon, after afternoon tea, on a little more mechanism and a few things like that. Look, it's a real pleasure to invite Jackie Batley. She's come from University of Western Australia all the way across here to be with us for today and tomorrow and the next day. Thanks very much, Jackie, for coming. And Jackie's going to talk about some Brassica disease resistance work. So thanks very much, Jackie.
Thank you, Christine. And it's a bit strange to be back here, actually, after 10 years. Walking through the campus, there's a lot that's changed. A lot that's familiar, but a lot that's changed. But it's nice to see some old faces as well. So yeah, I'm going to talk about the work that we've been doing in disease resistance and pangenomics and genomics, and talking about some of the struggles we've had along the way and how we've overcome them. So I work mainly on the Brassica species, and that's the examples I'll give today, although we have been doing some work in some other species. So the main species that I work on is oilseed rape or canola, which is a polyploid made up of two genomes of Brassica oleracea and Brassica rapa. And canola is one of the most important crops here in Australia, but it's also very affected by disease. And we get quite a lot of yield loss from these diseases. And something that I've noticed since being here in Australia for 20 years is that there's a much higher reliance on fungicides than we ever used to have when I first arrived in 2002. But the problem with that is that we're also getting fungicide-tolerant isolates coming up. So we need to have a way to control the diseases. So I work mainly on this disease here, blackleg, which is the most important disease here in Australia. And in certain years, we can get 100% yield loss. And this is particularly the case where growers keep growing the same cultivar with the same source of resistance. We can get, in a few years, there's been like complete devastation in some areas like the Eyre Peninsula. So I'm working to try and find genes to control this so that we don't have this reliance on fungicides and we don't have this big yield loss. And as I say, just a little bit about blackleg. We have a large yield loss in terms of monetary value. So this is why we want to be able to control it. And there's two different sources of resistance to canola. So there's qualitative resistance, which is major resistance. It's a single gene. It interacts with a gene from the pathogen, the AVR gene, and it offers complete protection from the cotyledon stage right through to the adult plant stage. And then we've got quantitative resistance, which occurs much more just at the adult stage, and it only provides partial resistance. And this is many minor genes. But what I'm going to talk about today is the work we've been doing to identify the qualitative resistance genes. Now, one of the fantastic things about resistance genes is that they have conserved domains, so they're actually really easy to predict within the genome. So a lot of people work on these nucleotide binding site leucine-rich repeats, but we also look at the transmembrane ones, the RLPs and RLKs. And although they have other roles, in brassicas and blackleg in particular, these are the genes which are mainly responsible. So we can do work predicting these genes within the genome just by looking at trying to identify these different domains. But we do have a challenge of resistance gene identification. The Brassica genome might be small. I can't complain because compared to wheat, it's nothing like that. It's a small genome, but it is very complex. And it's gone through lots of whole genome duplication and triplication. So for anything, we can have at least 6 copies. There's a lot of variability in the host and the pathogen. So we see between different morphotypes, different cultivars, we see a lot of variability in resistance. And one of the main problems that we have is that the pathogen is far quicker to adapt than we can get the plants to. So because there's this gene-for-gene interaction and the host recognizes a frequency of a virulence gene and can then protect the plant, what then happens is the pathogen population adapts and it no longer has that avirulence gene, and so then there's no resistance. So we need to make sure we can identify many of these different resistance genes so that we've got a lot of range of genetic variation that can be used and deployed in the germplasm. So going back to work that we started when I was here at UQ, we naively thought that once we had a reference genome, life would be simple and we'd find the genes and that would be it and we could move on to look at something else. So Salman was a PhD student here at UQ, and he did work predicting distribution of the resistance genes in the genome. And we can see, as I say, because there's many of them, we can see that each of these different symbols is a different gene class, and then we can see that some of them are clustered, some of them are duplicates where they're connected by lines. But we could predict where they were on the genome, and we were particularly interested in chromosome A7 because this is where a lot of the blackleg resistance was. So we did this preliminary work, and we have this, to explain what this is a little bit. So this is a chromosome A7. Each of these is different predicted resistance gene. And then we had, across the chromosome, about 60 predicted resistance genes that we could look at. And then we had a QTL region, which contained a few different blackleg resistance genes in it, which had about 30 different candidate genes. And then in one gene in particular, called RLM9, within that QTL region, there were only 14 candidate genes. So we did some whole genome resequencing of resistant and susceptible lines and mapped them to the genome and looked for SNPs in these candidate genes, thinking that this would be it, we'd find the gene. But sadly, none of them segregated for resistance. So then that's when we realized that reference genomes are great, but they're not the be-all and end-all. You can't, if the reference genome didn't have this resistance gene RLM9, we may never find it. So that was when we started to work on pangenomes. And this came about because we realized we'd been looking at this model here where we thought that there were genes and there'd be a SNP which caused the difference in the phenotype, and that that's what we were looking for. But then what we realized is that actually there can be genes which aren't present between different lines. So we've got structural variation, and maybe that's causing some of our phenotypes. So this led us to starting to develop pangenomes in the brassicas. We now have, and this is actually out of date, but my students did the slides and I didn't manage to get the new one from them. But we now have we have many reference genomes for the different species, Brassica species, but we also have pangenomes for many of them, and there is now a pangenome for Brassica carinata and Brassica juncea as well. So we've got this absolutely fantastic resource just in the 10 years since we got the first reference genome of more reference genomes, but also pangenomes. So we're using these to try and identify the resistance genes. And even just comparing differences between just the quality of the different reference genomes with the long sequence reads compared to the traditional one where we were first looking, we've got much better assemblies and many more genes. So we've got a much better resource to identify these resistance genes. But we started with a diploid. We decided to start simple rather than going for the polyploid because we didn't want to challenge ourselves too much. So we started, this was a joint collaborative effort with an international consortium of sequencing some different Brassica oleracea lines and creating a pangenome. So we didn't have that many compared to the pangenomes you get now, which have hundreds and hundreds of lines. We had quite a limited resource, but sequencing was still pretty expensive then. So we had an outlier of Brassica macrocarpa, but what we could see here, when we looked at the different Brassica oleracea morphotypes, these are different resistance genes which are in the pangenome, which weren't in the reference genomes which we had. The different colors just represent different gene types. But what you can see here is that the cabbage lines here had far more resistance genes than other morphotypes. And so if the reference genome wasn't cabbage, you'd be missing out on this whole resource of resistance genes. And this was just an example from Brassica oleracea and a fairly small number of lines. So by increasing the number of lines in a pangenome, you'd be able to find all these, you'd be able to find all these extra genes which weren't present in that reference genome. And we also found, we just did a bit of a study to look and see if there was any difference between these lines which were present only in the pangenome but not present in the reference genome. And what we found was that resistance genes which showed presence-absence variation, they had fewer SNPs than the R genes which were present in every single line. So there seem to be some different methods of evolution of these genes, whether they either get lost or they mutate to form resistance. We then moved to the Brassica rapa pangenome, and this had a lot more individuals compared to the 9, I think, which we had in Brassica oleracea. We had over 77 cultivars in the Brassica rapa pangenome, and these, again, were from many different morphotypes. So brassicas are really variable, and we managed to get many different lines from field mustard to heading cabbage to turnip within this pangenome. So we could really see what the difference was between the different morphotypes. And again, we could see that there's many more R genes compared to single reference genomes, and that in the reference genome, there were 365 resistance genes identified, and in the pangenome, there were 624. So there's this whole resource of genes which aren't present in the Brassica rapa reference. And again, we could just look at the density of them across the different chromosomes and just look and see whether there was any clustering. And we can see whether the density, you can look at the density of those which are variable. So sometimes we get large clusters of regions where the genes tend to show structural variation. And these are just examples of different reference genomes, and as the reference genomes improve in quality, you're starting to get more resistance genes assembled as well. But again, just like Brassica oleracea, we saw that the morphotypes hugely different in resistance gene number. So the field mustard had far more genes than, in this case, the heading cabbage or the rapid cycling lines. So this is really important for us when we're looking for disease resistance, because we need to be able to look at all of these and not just look at one single morphotype. And they also differ in the proportion of the different types of the resistance genes. So the field mustard has a lot more NLRs than the transmembrane ones. We haven't linked this to disease yet, but this is probably linked to specific diseases because some of them aren't within the cell. But we can also do comparative analysis between the diploids and the polyploids. So we could see Brassica napus has the A genome from Brassica rapa, and we could see that 93% of the resistance genes in the Brassica rapa pangenome were also present in the Brassica napus pangenome. So there has been conservation, and not too many have been lost. And we can compare this and see whether there's any difference, again, between the different gene types. So what's interesting is that Brassica rapa has a lot more NLR genes than Brassica napus, and Brassica napus has a lot more of these TM LRR genes. And again, that's probably linked to disease, because with blackleg, it tends to be the TM genes which are important. So we moved on to do resistance genes in the Brassica napus pangenome, which is what's important because this is here certainly the major crop. And this is Aria, who started doing an internship here but then moved to UWA with me. And again, this was with 50-something different cultivars. And Brassica napus tends to show a lot less diversity than some of the diploid species as well. So we were surprised when we found that 43% of the resistance genes in the pangenome weren't present in all lines. So we weren't expecting to see that level of structural variation in a species which generally doesn't show so much genetic diversity. But this would explain where we've got this many genes which aren't present necessarily in the reference genome. This would explain why we couldn't find our blackleg resistance genes. And again, we could look and see whether the variability came in on the A genome or C genome, or whether these were actually genes which weren't present in either of the reference genome. So some of these genes were present in the reference genome but showed variability in other cultivars, and these are ones which were present in other cultivars but weren't present in the reference genomes. And now we can do comparison between all the different pangenomes, so between Oleracea, Rapa, and Napus. And we've got many more individuals than those first pangenomes which we worked on. And so we can really now detect what presence-absence variation there is between all those different cultivars, but also the different species. So we can start to look and see the conservation between the different species on the A and C genome. And now take it wider, now that there's pangenomes for other Brassica species as well. We've started recently looking at Brassica carinata, which is a mustard, Ethiopian mustard, just to look at the differences which we had in the genomes, because we wanted to be able to compare the C genome to Brassica oleracea and Brassica napus. We could see, again, this is in the B genome and the C genome, so we've got clustering of the genes. We've got whole regions where there's no resistance genes at all, which is interesting in itself. But we can start to look at the total number of genes in the two different genomes. And what we see here in carinata is that we see that a lot of these genes, which are showing structural variation, a lot of them are duplicates. So we're seeing copy number variation of some of the genes as well as gene loss. And interestingly, we found that there's subgenome dominance in Brassica carinata. So we're finding that the B genome is preferentially retained after homologous exchanges, and that the C genomes experience more genomic rearrangements. So it's interesting to look at the polyploids and maybe see what the selection pressure is for some of these genes. Again, we did comparative analysis between the two diploid progenitors of Brassica carinata, and just looked to see the difference in the conservation. We've got the Brassica carinata genome and the Brassica nigra genome. And we can see that we've got, they're not always, the gene copies aren't always on the same chromosome. There's been quite a few rearrangements for some of them, even if the genes are still present. And what we found was that Brassica nigra has more genes than the Brassica carinata B genome, but the Brassica carinata C genome has more R genes than Brassica oleracea. This could be partly due to cultivation. We don't know, because Brassica nigra hasn't actually gone through that much breeding, but it's something that we want to look at a little bit more. But we found that there's different evolutionary patterns of the resistance genes. So it could be expansion of some of them, so we get other copies. It could be straightforward inheritance, so they just go from the B genome, for example, from nigra to carinata. We see sometimes that the R genes are inherited in favor of one of the diploid parents, so either the B nigra or the B oleracea, and the other one's lost. We see loss of R genes in Brassica carinata compared to the diploids, and we quite often see rapid sequence diversions. And now what we want to do is be able to link all that to actually specific diseases, because that's where it's important. But we've also found that copy number variation is important. And this is an example of a circus plot in Brassica napus. Each of these is a different chromosome. And the blue is where there's deletion. So each of these inner rings is a different cultivar. The blue is where there's been deletion of a gene, but the red is where there's been extra copy numbers. So we can see that for the resistance genes, there's both duplication and deletion of them. So we're seeing quite significant structural variation. And when we look at the specific genes on the different chromosomes, we've got some which show loss in some lines, some which show at least duplication in some lines, which is the blue, and some which can either be lost or have multiple copies. But what we're interested in is looking at them in known disease resistance QTL. So when we look in Brassica rapa, we've got all these extra lines, genes in the pangenome, which weren't present in the reference, and many of these sit within disease QTL. Similar for Brassica napus, and this is where we're seeing that there's a lot of these candidate resistance genes sit within blackleg QTL. And the success story of that was that going back to that original work 10 years ago where we thought we had a reference genome and it would be easy, Yuki and Tina worked on these genes RLM4 and RLM7, and through a lot of different analysis which they did, they found these genes in the pangenome but not in the reference genome, and we never would have found them otherwise. So now we have these genes where we can develop markers which we can deliver to industry, so we've got Cas markers for these, and we're developing a few other genes as well, because there's actually quite a few of them which haven't, 5 have been cloned, but we're now working on many of the other genes now that we've got this resource in the pangenome. And we can basically now work on some of these other genes which are coming from more wild species. So what we've realized is there's very little diversity within the germplasm of Brassica napus that's been cultivated, but there's resistance in other wild or subspecies which can actually be integrated into the napus genome, which we can use for identification of resistance genes. And this is a really important source of diversity and could be a reason for some of the variation we see between the different cultivars, because there has been a lot of this integration work going on. So we've now been working on 2 other genes, LEPR1 and LEPR2, which come from Brassica rapa sylvestris, and then a gene RLM6, which comes from the bee genome. And we can go through and map these onto the different pangenomes and find some significant associations. So this is one of the LEPR genes, and this is the bee genome-derived gene. And we can go through and compared to just what we had before on the reference genomes, we can now look at the QTL in a pangenome and identify a much larger region and many more candidate genes, which we can look at to try and see whether they are the causal gene. And then we resequence them between resistant and susceptible lines and look for conserved resistance alleles. And we've managed to very quickly, just in the space of a few months, go down to identify just a few candidate genes. There's 1 for that gene. We've got 2 for those, which we've now actually narrowed this down to 1 of these genes, which we've developed markers for, which we're using for breeding for resistance. They're being developed. And so we can see that there's a lot more in the wild relatives that we want to bring in a little bit more. So we've started doing some work looking at characterizing resistance genes in the Brassicaceae. So Sude did this work, this is unfortunately just in reference genomes because that was all that was available at the time. We unfortunately don't have many pangenomes for any of the wild species. And so she took the reference genomes and did resistance gene prediction in these genomes. And you can see there's each of these, again, just the different colors represent different types of resistance genes. And you can see that there's quite large variability between different species within the Brassicaceae. But then there's some things that caught our eye because didn't look quite right within some of the analysis. And so there's a few, let me find the right one. It's actually probably arrow on the next slide. So what we found was that there were some cultivars from the same species which showed vast differences in the number of resistance genes. And what actually attracted us a bit more to looking at this was that some of them were actually new reference genomes from the same cultivar, which had just been developed with different sequencing technology. So this made us realize that predicting genes in genomes and pangenomes is very good, but a lot of it is very dependent on assembly and annotation of those resistance, of the genomes. And you can only go so far with that. So we wanted to look at this in a bit more detail to try and understand why we were seeing such differences within the different cultivars within a species, but also between different assemblies of the same cultivars. And so Philip did some work. He was a postdoc in the group, and he did some work looking at this. And what he found was that different repeat maskers were being used for different genome versions, different annotations, and so they were actually repeat masking out the resistance genes from the reference genomes. So he managed to reverse engineer this and pull the genes back in, and we could actually then, now with all our genomes, we go in and make sure that we're not losing genes due to repeat masking. But the one good thing that we did see from this was that in our pan-genomes, we weren't seeing any difference due to this. That was truly due to structural variation, and we did quite a lot of work to validate that. So in conclusion, we've got a lot of rich genomic resources now for brassicas, and it really does allow faster identification of genes. From the first genes that we tried to identify for black leg resistance, which took us probably 10 years, to ones now where PhD students are doing it within just a few months of their PhD. But what we do see is that structural variation plays a large role in resistance. And one thing I didn't show with the pan-genomes, I showed all the disease resistance genes we saw which showed structural variation, but they aren't the only gene classes which show significant structural variation. So this isn't just important for resistance, it's important for other traits as well. But we see that a single reference genome is not enough, so I really want to try and get more pan-genomes, not only for the cultivated species, but also for the wild species, so we can see what resources we can bring in. All these improved tools and genomes are really accelerating the breeding. It's really nice to be able to see the markers being used and deployed in breeding programs. And then we can also look at, while we characterize the R genes, we can also look a little bit more at their evolution, start to look at the selection pressure of some of them and link them to disease where we haven't been able to do that already. But I'd just like to thank, obviously, my funders, GRDC and the ARC fund most of this work, and also the members of the group, because they're actually the ones who do the work. I just come around and present it all on their behalf. Thank you.
Thanks, Jackie. That was quite amazing. Thank you. Any questions? There's a question there, Barbara.
Sorry. So you're saying, I think, that the genes that had presence-absence, the R genes with presence-absence variation had a smaller number of SNPs. So I was wondering, was that like synonymous and synonymous SNPs, or just sort of one or the other? Like, is it like that they're just younger, or is it that they're under increased constraint?
Now, I can't remember because we looked at that quite a few years ago. I think it was both, but I'd actually have to go back and check.
Yeah, cool. It was really interesting.
Yeah, no, we published that a few years ago, and of course, yeah, I can't remember last week or more.
Peter?
So I was wondering, do you see when there's recombination, where we have a region and you're saying it's duplicated, that it mimics like a human immune system, that you actually see these genes sort of recombining and creating new variation, or just what you've got is what you've got?
I don't think we have seen that so much. I think it is just what you've got is what you've got.
That's a shame.
I know.
More questions? We are right on time, so thank you very much, Jackie, for the presentation.